The Parkinson's podcast is brought to you by the Davis Phinney Foundation and brings you the stories, wisdom, and expertise of people living with Parkinson's, care partners, and Parkinson's professionals. I'm your host, Sia Euros, and in this episode, I'll be talking with Roger Barker, Professor of Clinical Neuroscience and Honorary Consultant in Neurology at the University of Cambridge and Addenbrooke Hospital. Dr. Barker is known for his groundbreaking work combining basic research on novel therapies for chronic neurodegenerative disorders like Parkinson's, with clinical studies aimed at better defining these conditions. This episode is sponsored by Blue Rock Therapeutics, a clinical stage cell therapy company developing cell therapies for multiple conditions, including Parkinson's. Hi, Roger. Thanks for joining us today to talk about cell replacement therapy for Parkinson's. Before we dive in, could you tell us a little bit about yourself?
Yeah, so I'm a neurologist in Cambridge, UK, and I started working in this field, 35 years ago, I think. And what I was particularly interested in was the idea that you could repair brains. And in Parkinson's, as you're well aware, there's a loss of a very particular population of cells, these dopamine cells. And you lose a couple 100,000 and you get the first motor features of Parkinson. So it seemed like a very obvious target to try and think about repairing the brain with it. So I came to Cambridge so I did my medical degrees in Oxford and London and then my general medical training. Then I came here to do a PhD around trying to work out how we could start to repair the brain. That was all very much non-human studies. And then out of that, I developed a very great passion to see if we could translate that into the clinic. And till really the turn of the century, that was all done not with stem cells, because there was no stem cells which you could adequately turn into dopamine cells. But in the last 10 to 15 years, this has gained great prominence. So we've been using stem cells within a consortium within Europe, and I very much take the clinical lead on that, to try and take stem cells, turn them into dopamine cells, and put them into the brains of people with Parkinson's. So we're 3/4 of the way through a trial. Prior to that, we did a different dopamine cell therapy in people with Parkinson's. So this has been an area I've been very much involved with for 35 years. And actually, next month, September 2024, it's 50 years since the first experiment was done on trying to repair the brain using transplants of tissue by Anders Bjorklund and his team in Lund, Sweden.
And so what exactly is a stem cell, and are there different types of stem cells?
Yeah, so stem cells essentially have this unique quality that they can divide, so they can reproduce themselves, if you like, and then they can differentiate into a different type of cell. So in theory, a stem cell can carry on reproducing itself forever. And then as it reproduces, some of its progeny, some of the cells which are divided out of it, differentiate into a different type of cell. And so every single one of us, both here and listening, began life as a stem cell. We all began life as an embryonic stem cell, because that is the fertilized egg, turns into embryonic stem cells, and they obviously give rise to every single cell in your body. So there are stem cells which are there in normal development. There are stem cells which you can make by taking cells and reprogramming them, putting them back into a different state. There are stem cells in our bodies now. So we have stem cells in our bone marrow. So our blood gets turned over frequently. They are stem cells. We have a type of stem cell in our gut. They keep turning over, and in the liver. So there are different types of stem cells. Some have the potential to turn into any cell type, like an embryonic stem cell. Some are rather restricted in what they can do. So obviously, if you're in a bone marrow, inside the bone making blood cells, it wouldn't be clever if they could go off and make any type of cell, because you just want them to make blood cells. So they're more restricted in what they can produce. But yes, there are many different types, and it causes confusion because people use the term stem cell as a shortcut. And actually, it can cover a whole multitude of different cell types, which have very different properties and very different characteristics. And especially in the world of Parkinson's, there are many therapies which are offered around stem cells. And you really need to dig down to find out what that stem cell is and exactly what it is that people are trying to do with it, to understand what the purpose of it is and what they're trying to do with it with respect to Parkinson's.
And so why might stem cells in particular be useful for treating Parkinson's?
Well, the great advantage, as I've said, is they can reproduce. So you can produce vast numbers of them. So that's great because you can make, in theory, an infinite number of cells. So you have a great supply of cells. The question then is, can I turn them into the cells I need to treat people with Parkinson's, and the obvious one would be dopamine cells. So they're very attractive in the field of Parkinson's because we could get a stem cell and we could convert it into a dopamine cell, and then we could transplant it into the brain of people with Parkinson's to try and repair that which is lost at the heart of the disease. They can also be used in a slightly different way, because you can obviously use that same technology to say, well, could I then study the dopamine cells in the lab? So could I get a stem cell? Could I make a stem cell from a patient, turn that into a dopamine cell, and then study it in the dish and say, I'm now looking at that patient's dopamine system, which I can't really see in their brain, but I can now see it in a dish. What's going wrong in their cells? Why is it going wrong? And how can I make it better? And so you can also use stem cells to study disease in this condition. So you can use it in both ways. And obviously there is a little bit of a tension there, because if the cells can develop the pathology of the disease you want to look at, why would you transplant them into the brain if they're from the patient themselves, because they may develop that pathology? And we may come on to discuss that. But essentially, stem cells for disease, use it as a therapy to repair the brain, or use it in the lab to study what's going wrong in the condition itself.
So you can use your own stem cells and stem cells from others in treatment? How does that work?
Yeah, so basically, I would say there are two major types of stem cells that we tend to talk about in this area. One are so-called embryonic stem cells, ES cells, and these are the fertilized eggs. So, when the egg gets fertilized, it starts dividing. Those are the embryonic stem cells that give rise to every cell in the body. If you can harvest those cells from embryos from IVF programs, bare embryos, you can turn those cells into a cell line and you have an embryonic stem cell line. Now, those lines are essentially can be turned into any cell that you want, but they clearly don't come from someone with Parkinson's. There are lots of ethical issues, obviously, with using embryonic tissue, because you would have to destroy the embryo in order to make the line. So it's not an area that's not without controversy, and especially in the United States and in other countries around the world, this is a very hot topic. But nevertheless, you can still use embryonic stem cells to study disease and to treat people. The other major type of stem cell is a thing called an induced pluripotent stem cell. So this is what Shinya Yamanaka won the Nobel Prize for a number of years ago. And in essence, what you do there is you take an adult cell, so a skin cell, a blood cell, and you then reprogram it. So you take that adult cell and you say, I want you to look like an embryonic stem cell. So you put in a number of factors to reprogram the cell back to a stem cell. So it looks like an embryonic stem cell, but it's obviously now come from the patient. And then you drive that in the direction that you wish to drive it, in this case, dopamine cells. And so in that sense, you can then study disease from a patient by using the patient's own cells to make stem cells, to make dopamine cells. You could also use that technology to treat the patient themselves. So take a bit of skin, say, from a patient, turn it into induced pluripotent stem cells, turn it into dopamine cells, transplant it back in. That has some issues because they've obviously declared already that they're not very good at keeping dopamine cells alive because they've got Parkinson's. But you could also use it for transplant from somebody who hasn't got Parkinson's. So take someone who's an adult who hasn't got Parkinson's, take a bit of skin, turn it into stem cells, turn it into dopamine cells, and then I use that as a way of repairing the brain in someone with Parkinson's. So induced pluripotent stem cells have much less ethical concerns, if you like, because they come from an adult cell. There are other issues with it about, obviously, if they come from the patient themselves, about disease developing in that cell itself. And obviously, you've had to reprogram that cell, whereas an embryonic stem cell is just doing what it does. It's going to turn into those cells anyway. So you're rather encouraging it to follow a fate it's due to follow. Induced pluripotent stem cells, you're engineering it and forcing it to become something that it wasn't planning to. And that obviously might create some concerns about safety and such like. But those are the two major types. As I say, there are other types of stem cells, but those are the major ones I would say POGs that people should concentrate on. And others I would tend to ignore, like mesenchymal stem cells, adipose, fat stem cells and such like.
When you say reprogram, what do you mean by that and how does that happen?
So basically, one of the things that happens is that your cell, you start from a stem cell and then it slowly commits itself to some sort of fate. So you begin life as an undifferentiated student, and ultimately you either end up doing podcasts or you end up being a doctor. And so you differentiate into that state. Now, obviously, if I wanted to go back to be a teacher, I would have to be reprogrammed to going back to be a teacher. So you would have to do something to me to get me to be a teacher because I'm now committed to being a doctor. And it's the same sort of principle, basically. So your skin cell has decided to become a skin cell. It began life as an embryonic stem cell, but it's now turned into a skin cell. So you have to say, I don't want you to be a skin cell. So I'm going to have to switch off the things that make you a skin cell. I have to re-express the things that take you back so that I can switch you into any direction you like. So you reprogram it back into a more primitive state. And to be honest, when people sort of started thinking about this, you think it's a bit crazy. I mean, could you really do that? And when it was actually first done, there was a certain amount of skepticism as to whether this really could have happened with just using 4 factors, which what they originally used in their studies, by Yamanaka factors, and it proved to be the case. So it's really taking away the identity of the cell that it has, the adult cell, and putting it back into its primitive, how it began life as a stem cell.
Is it like a chemical process, or like, how does that happen? Does it happen in the lab?
So it happens in the lab, and different people have slightly different ways of doing it. Essentially, the idea is, you, as I say, you have to re-express those bits of the DNA which make you into a primitive cell. And the key is to switch off the things which commit you to something being some other fate, really. So there are different, they're often called transcription factors, but there are other ways in which people have done it. But essentially, the idea is to convince your adult cell to stop being an adult cell and to go back and be a very young stem cell. So you rejuvenate the cell. So obviously then people start saying, well, if I can do that, can I rejuvenate everybody by using something similar? So some people are very keen on that. But that's slightly, we're getting slightly off piece, and I'm not entirely sure that we could really do that.
So how were stem cells first used, and what was that like for, the first person to receive this kind of a treatment?
So in terms of stem cells, we're still a little in the early stages of it, but the very earliest transplants that were done were back in the 1980s. And so the original transplants were not done with stem cells at all. In fact, they weren't really done with dopamine cells. So the very first transplants were done with tissue called the adrenal tissue, which is a gland that sits near your kidney, hence the name adrenal, by renal kidney. And the adrenal gland produces a lot of hormones, but particularly adrenaline, noradrenaline, and things of that nature. So when you get a bit excited, that's what pumps out a lot of these hormones. And it was found that they produced a little bit of dopamine. So people tried to transplant the adrenal gland into the brain of people with Parkinson. So the idea was, if it produced a bit of dopamine in the tummy, that's not much use to me. If it produces a bit of dopamine in my brain, let's do that. They took it, so it's called so-called autologous because the patient provided their own tissue. They basically just transferred their adrenal from their tummy into their brain. And that didn't really work because the tissue didn't really survive and it didn't produce much dopamine. But whilst that work was going on, the team in Sweden, particularly Anders Björklund and his clinical colleague Ulla Lindvall, said, well, we're working on the developing dopamine cells. So the dopamine cells you lose in Parkinson's come from a bit of the brain called the midbrain. And in development, that's called the ventral mesencephalon. That's the developing bit of the brain that makes the dopamine cells, which eventually form this pathway, which is what's lost in Parkinson's. And they were transplanting in fetal dopamine cells, so developing dopamine cells into the brains in rats. And they said, well, these cells do very well. Why don't we do the same with human dopamine cells? So and that's obviously pretty ethically controversial because the only way you're going to get developing fetal dopamine cells from humans is getting them from human fetuses. And the only way you can do that is from termination of pregnancies and abortions. Anyway, in the late 1980s, this work began and a number of other centers around the world took it on. And what it showed was in those early studies that when it worked well, it worked extremely well. So the most successful patients who've had fetal dopamine cells, so these are not stem cells, transplanted into their brain, they have normal dopamine levels in their brain on imaging. So you can restore dopamine levels back to normal in the brain in people who have vastly deficient dopamine levels because of Parkinson's disease. So in these patients, some patients you could restore it back to normal. They came off all their medication, so they didn't require any more dopamine drugs. And that worked up to 20 years, 25 years from the transplant, which was 30 to 35 years after they first developed their features of Parkinson's. So when it worked well, as a proof of principle, it showed that if you put fetal dopamine cells into the brain, you could put that part of the pathway back to normal. And in some patients, you could essentially return it back to normal. Now, it's not a cure because they still have the disease going on in the background. But to all intents and purposes, it is a cure because you've essentially got rid of the problem which they were complaining about. Then the question is, well, why isn't this everywhere now? I mean, if it's so crackerjack and works fabulously well, why isn't everyone having it? And the problem was, outside of the ethical problems, there were clearly logistical problems because you needed a lot of fetal tissue to transplant any one patient. And it was quite clear that it was very inconsistent. So I've told you about people who went entirely back to normal, in essence. There were other people where it made very little difference. They had to carry on with their drugs and eventually they progressed and needed deep brain stimulation. And some of the patients, especially in the studies that were done in the United States with this approach, ended up getting side effects from the treatment, was that the transplant generated abnormal movements in the patient itself. So everyone who's on L-DOPA, So in the UK, that's Sinemet and Madapar. I don't know if it's the same in the States, but these are standard L-DOPA drugs, been around since the late 60s. Everybody with those drugs after a number of years starts to develop some involuntary movement, so-called dyskinesias. And they can be very troublesome and you can have to manipulate their medication. But in some of the patients with transplants, they develop these dyskinesias in the presence of the transplant and the absence of the drugs. And that's a bit of a problem because obviously I can stop anyone's drug-induced dyskinesias because I can stop their drugs. They might not want me to, but I can. If you've got a graft-induced dyskinesia, I can't get your graft back. So now you're in trouble. So some of the early patients were very important for proof of principle, showing when you get it right, it transforms what you can do. But clearly it's inconsistent. And so trying to work out what that inconsistency is, whether you can control it and then develop a better therapy around the principle means it could, in theory, then become a sort of standard of care if you could get it to work consistently.
And so what are the theories behind why or why not it would work?
So there were a number of sort of thoughts, really. And if you sort of go through each stage, so one, I think in the 1980s and 1990s, there was, this isn't strictly true, but there was a sort of perception that Parkinson's was a single entity. If you had Parkinson's, you had Parkinson's. That was it. And actually, I think understanding there are subtypes of Parkinson's and some people follow different courses. So trying to understand what we call the heterogeneity, the variety of Parkinson's was important. So it was probable that there were different types of patients who were transplanted. You have to remember in the 80s and 90s, there were no genes staying for Parkinson's. The first gene for Parkinson's was discovered in 97, 98. So there were no genetic forms of Parkinson's recognized. There were no different subtypes. So I think part of the problem was that the patients were all a bit different. So there was no consistency in the patient that was chosen. Secondly, I think there was a problem in terms of the stage of disease. So naturally, when you do these experimental therapeutic approaches, you tend to use people who are more advanced with their condition because they have less to lose, if you like, and that might not necessarily be the optimal group. So I think there were issues about type of patient and stage of patient, and linked to that was age of patient. So the older patients seem to do less well. So that was one problem, I think, was around being better at selecting patients or realizing that patient variability was driving some of the inconsistent results. I think the second was that the amount of tissue which was put in varied greatly between trials. So in essence, you've got to get about 100 to 200,000 surviving dopamine cells for it to work. And when you transplant cells into the brain, not surprisingly, large numbers of them die. So different people use different amounts of fetal material. So I think the survivals were very different. And that related in part to how you prepared the tissue, how much tissue you put in. And then the other key thing was the use of immunosuppression. These cells which you implant are not the patient's own cells. So you will recognize them as not being your own. They'll be a bit foreign. And as with any foreign tissue, such as a bug or a virus, you'll try and kill it off because that's what you're designed to do. And so you will get a bit of an immune response to this. Not much, but you probably get some. And so some people thought that was a big thing. So let's stop it with immunosuppression, which you would give if you were having a new heart, a new kidney or whatever. Some people thought not. So I think there were problems there about surviving cells related to how much tissue you collected, how well you prepared it, how much immunosuppression, how you delivered it. And then the sort of final issue, I would say, was around follow-up. So one of the interesting aspects of this whole approach is you're putting in new cells which are not dopamine cells. They're destined to become dopamine cells. So they have to first of all turn into a dopamine cell. They then have to connect to the brain of the person you've put them in, who has to connect to them. You then have to some extent, slightly learn to use these new cells. And that system needs to maturate. So that probably takes 2, 3, possibly 5 years. So obviously, if you go too early and say, what do I see at 6 months? You can often say, well, it hasn't worked, waste of time. But of course, you haven't left it long enough in order to really work. So I think a lot of the inconsistency, as I say, was around these issues: patient selection, tissue preparation and delivery, follow-up and immunosuppression. We attempted to try and look at that at the early part of the century when it was all slightly unraveling in the face of some trials to try and identify those factors, which we then took forward into a new trial. But ultimately, the use of fetal material is never going to be possible as a therapy outside of the ethics, because the logistics of it, you simply cannot get enough tissue to treat all the people who would benefit from it. And more fundamentally, you can't standardize it. It's not a manufactured product. So what you're doing is you're collecting the fetal material, you dissect, you prepare the cells, but that's not standardized. You can't standardize because obviously the tissue source is not from a unitary single source, which is where stem cells are very attractive. because I can make stem cells and then I can just manufacture a vial of dopamine cells from them, which will be the same in every person who gets it. So stem cells offer a lot of potential advantages over anything we can do with fetal tissue, just around ensuring that everyone gets the same product, which should reduce the variability. It doesn't get away from the patient heterogeneity, these different types of patients, but it does get away from some of the other problems with preparing the cells.
So are people working on a solution for this? Are these being artificially grown? Is there a science behind that?
Yeah, so what most people do now, and I would say this really took off in about 2011, 12. So there were two groups in the world that sort of cracked the mechanism by which you could persuade a stem cell to come into a dopamine cell. And people often say, well, why did it take so long? And you think, well, if I was an embryonic stem cell, I'm going to make your body. You've got 500,000 dopamine cells on each side of your brain. So there's 1 million cells in your body that are dopamine cells. I mean, why would I want to be a dopamine cell? I mean, that is a tiddly number of cells compared to all your skin cells, liver, gut, all the other brain cells. So, I mean, you wouldn't want to become a dopamine cell. So you've got to obviously work out what is needed to persuade it to become a dopamine cell. And so that was discovered by the group of Lorenz Studer in New York and Marlin Palmer in London. They worked out that the developmental pathways which drove stem cells to give rise to dopamine cells, it wasn't thought to be as it was in mice. So they suddenly had to rethink the whole thing. But they worked it out. And actually, one of the reasons this has become quite attractive as a therapy is the process for turning a stem cell into a dopamine cell is quite quick and relatively easy. So having said it's quite difficult to make it, actually once you crack the code on how to do it by putting in different factors at different times, it's actually relatively easy. So you can make dopamine precursor cells which will become dopamine cells from a stem cell within 16 days. So 16 days in the lab, take a stem cell, convince it to become a dopamine precursor by various protocols, adding things and persuading it, and then you're away. So if you think the major cost for delivering a therapy is manufacturing, it's a very efficient process from a little 6 well plate, which is 6 little wells where you grow cells in the lab. If I did that myself, well, it would be hopeless. But if someone knew what they were doing about this, they could make enough dopamine cells from 1 6 well plate in 16 days to treat 500 patients. So if I had 1,000 plates and a robot, I could treat 500,000. That's North America sorted, one manufacturing run. So this is why it's slightly caught fire in terms of recent years, because I think pharma has seen that there is great potential in this, because actually manufacturing a standard product is not colossally expensive. And obviously the shorter the manufacturing run, the more likely it is the product's always going to be the same. Whereas if you've got to grow something over 150 days, it's much less likely to end up the same after 150 days, because there's so many little things that could go wrong and differ. So it's very efficient, it's very short, and therefore the cost is relatively low. And it means you can make a lot of cells which you could use and make a profit from it, which is obviously what they need to do. But you would have a consistent product which everyone would have the same.
And so what's the difference between stem cell therapy and cell replacement therapy? Are they the same thing?
Well, it depends the context, really. Obviously, stem cells are, as we've discussed, anything that has this potential to continue to divide, and then you can persuade it to become some different type of cell. So stem cell therapies could be used for Parkinson's, they could be used for diabetes. You make the islet cells that make insulin, could be used for repairing the heart, you make heart cells, could be used for skin grafts, turn them into skin cells. Any part of you could be open to a stem cell therapy. So stem cell therapy just refers to a whole area. In the context of Parkinson's, it's generally meant to mean that you're looking at dopamine cells, and cell replacement is obviously what it says on the tin. You know, I'm replacing the cells that are lost, so it's dopamine cell replacement. Now, the problem is different people have different approaches to stem cell therapies, many of which don't have a great scientific basis. So people will talk about stem cell therapies to treat Parkinson's disease by injecting cells, say, under the skin or into the circulation, and they are supposed to work. Quite how they work, I don't know how they will work. I don't know how they get in your brain. I don't know how they're going to change anything. So that is not cell replacement. That is a cell therapy. They're often stem cell therapies, and often the mechanism is extremely unclear. So I would say in Parkinson's disease, the only credible way at the moment, and the only sensible way about talking about cell therapy, is to talk about stem cells and cell replacement around dopamine cells of the type lost in Parkinson's. That's really what I think people should restrict their discussion to, and not get distracted by other types of stem cell therapies.
And so how many people with Parkinson's have been treated with cell replacement therapy?
Oh, that's a difficult question to answer. So I would say with fetal tissue, I have no idea actually, but I can put some guess at it. I would say with fetal dopamine cells, I would probably say it might be a few hundred. So, I mean, in the Swedish group, they did 17. In the two NIH-funded studies in the States, there were 33 and 23, I think. So the numbers are always a dozen in all of the studies, really. So probably by the time you've had it all up, it might be a few hundred, if that. These are what we know about, which are being published and are working around this principle of using fetal cells to replace dopamine cells. If you then extend that to stem cell dopamine therapies, then the numbers are much smaller. So currently, the number of people who've had a stem cell derived dopamine cell for Parkinson's disease, there's one that's been published from the Mass General, which was appeared in a journal called the New England Journal of Medicine about 3 years ago. That was a single patient. As far as I know, they've not done any other patients. There's a company in California called Aspen Neuroscience, which again uses the patient's own cells to make dopamine cells. So they use so-called autologous induced pluripotent stem cells. They started this year. I don't know how many they've done, but I imagine it's 2, 3, something like that. The group in New York, Blue Rock, have done 12 patients. The group in Japan have done 7 patients. We have done 6 patients. There's a group in South Korea that have done 12 patients, and there may be a few groups in China that have done something. So overall around the world, it's probably less than 100 have had stem cell derived dopamine cells, and fetal dopamine cells, it's probably a few hundred. And if you look at all the clinics around the world that offer stem cell therapies, which are not around dopamine replacement, there's probably hundreds and thousands. So this is what we call stem cell tourism. So which is a very different area to what I'm really talking about.
And so what are the current clinical trials that are in process or in progress right now?
So the main trials at the moment are, so in Japan, as I say, they've done a trial with 7 patients, so Jun Takahashi is the person who leads it out there in Kyoto, and they're heading forward with new trials. They will probably do some in Japan. So Japan has a very different regulatory landscape to the rest of the world in terms of these type of therapies. But the company behind what they're working with in Japan have now filing to do trials in the United States. Aspen Neuroscience in California have funding to take patients' own cells, turn them into iPS cells, turn them into dopamine cells. So they're starting. There are a couple of other companies which are setting up in the States. The group in Blue Rock, which was Vivian Tabara and Lorenz Studer's group, which are part of Bayer, they've done 12 patients. They're now planning their next trials. We're doing our trial, but we work closely with a company called Novo Nordisk, who are also planning to develop this therapy up. I'm not quite sure what's happening in South Korea and China. So at the moment, the trials are all very much in the early stages. So if people ask, which is often a question I get asked in clinic, as was this morning, or on email, can I join your stem cell trial? The answer is no, because they're very limited at the moment. But they are happening. And I would say the most exciting thing in this field at the moment is that to date, there haven't been any big problems that we've seen in any of the trials, and that there are major companies involved with this. So Blue Rock are part of Bayer. Our work is linked to Novo Nordisk. So these are some of the biggest pharma companies on the planet. So the reason that's so exciting is that if this starts to produce positive results, and at this stage we don't know because it's, the first patient they've been transplanted in the last, few years, and there's still lots of things we need to sort of optimize. But if it does look promising, these companies have the capability and the expertise to push this rapidly to market, which is a big difference from the world of academia. So if I do a trial and it works, I've then got to go off and find another stack of money to do the next trial. That works, I've got to find someone to invest in me and take it into market. So it takes forever. Companies, this is what they do. So we sit at a very different place to any other time with cell-based therapies, dopamine cell-based therapies, because there is big money behind this. As I say, if it works, it will rapidly translate to the clinic and transform what we can do.
What are the theoretical safety risks of cell replacement therapy?
Well, there are a number, really. And I suppose if we start with the product, so obviously if you've got so what I said about a stem cell is it can divide. So that's terrific, because that's how you can make lots of cells. Obviously, if it starts dividing in your head, that's not such a great and clever thing for it to do, because ultimately, tumors and cancers are cells that continue to grow and you can't stop them. So the biggest safety concern, and obviously it's what the regulators worry about, is these cells turning into tumors and the stem cells continuing to grow. Now, to date, we do not see that. So we've never seen this in our preclinical work, so the lab-based work on these cells. And obviously the numbers of patients are very limited, so we can't say it never happens, but it hasn't been seen at all. And the reason for that is twofold, really. One is that as you persuade a stem cell to become a dopamine cell, all of the conditions you use to turn it into a dopamine cell are the very conditions which stem cells don't like. So to convince it to become a dopamine cell, you're growing it in conditions where stem cells say, hey, I'm out of here. It's just not good for me. So you're actually killing them off. And then obviously what the regulators wanted, as did everybody else, including ourselves and clearly, people with Parkinson's, is to screen the product to show that it has no stem cells in it at all. And so with modern technologies, you can be pretty certain you've got no stem cells in there. So you're transplanting cells which originally were stem cells, but what you're transplanting are not stem cells. They're cells committed to dopamine cells. The stem cells have died off. And so whilst there is a safety risk, we feel it's minimal. Now, obviously, the gamble when you do this in the clinic is that in animal experiments, a long-term animal experiment is 9 months, a year. You want to know, are these cells safe after 5, 10, 15, 20, 25 years? The only way you'll ever know that is by doing clinical trials. We're as convinced as we can be there's no risk of these stem cells coming back and growing. But, we won't know until we've done these trials. I think we're all confident that won't happen, but the proof will be that's what we see in the clinic. So that's one of the safety risks. The second, there are a number of other safety risks. One is if the cells decide to go walkabout, you could be in a bit of trouble. So, if it decides to go and potter off into another part of your brain, you think, well, that's not a great thing to do. A lot of the work we do is in mouse and rat brains, which is pretty tiny compared to the human brain. So if they migrate in the rat or mouse brain, in a human brain, that is essentially means they're just migrating in the same area where you've put them. So the way we do it at the moment, and I think will be the way for the indefinite future, is that the cells are injected into the site where you want them to work. So they're directly injected into the brain. And what we know is if they're directly injected into the brain, they tend to sort of stay together and stay in that site. There's no evidence we've got that once they've been implanted, you get a bit of reflux, so they go back up the needle tract where you put the needle in a little bit, but we try and delay taking the needle out so the cells don't go back up. They tend to stay where they're put, really. So as much as we know, these cells do not have the capacity to migrate. They stay where they quite like each other, so they tend to stay together, and then they grow out and innervate the area where they're placed. And what's extraordinary about these cells is that they seem to know what to do. I still find it extraordinary that they don't just sit there and pump out dopamine. They actually connect to the brain and the brain connects to them. So these are not just little dopamine factories. They are restoring, to some extent, circuits in the brain. And I guess, coming back to the original studies, one of the extraordinary aspects of that work is that the fetal material you transplant into the brain obviously comes from termination of pregnancies, abortions. That tissue is dead because it's obviously been aborted. So if you just take the sort of basic principle and say, I'm going to get a bit of dead fetal brain tissue and stick it in the head of an adult person who's got a ongoing brain disease, what would I expect to happen? Well, it would just die. Why would I ever expect that to live and do anything? And the extraordinary thing is it does. And so I think apart from the extraordinary aspect of that, it also shows that the human brain is incredibly good at adapting to pretty crude repair processes. So if I just got my car and it had a flat tire and I sort of threw a tire at it and sort of say, well, hopefully that will do the job, you'd think very unlikely. But actually, in some ways, that's what our brain repairs like. You put these cells in, they seem to know where to go. They seem to know how to connect. You seem to know how to use them. And they like to be where they want to work. So I think whilst there are safety issues about migration and other things, actually they seem to have an amazing capacity to work in the network which they're supposed to work in. They seem to have learned that developmentally. And so as they grow, they stay where they want to be. There's obviously the risk of surgery itself because these cells have to be implanted into your brain. So they have to be injected into the brain. So there is the risk of surgery, bleeding in the brain, things of that nature. And then at the moment, for most of the trials, we're giving the cells which are not the patient's own cells. So we have to immunosuppress the patient. So there are risks related to being on immunosuppressive drugs. Now, unlike kidneys and hearts and such like where you stay on these drugs forever, with these cells put in the brains, we typically only leave people on the drugs for a year, where the risks are actually relatively small. So there are risks from the cells themselves, there's risks from the surgery, and there's risks from the immunosuppression. And one of the challenges, and I think one of the slightly underplayed aspects of clinical trials, is the bravery of the patients who go into this. Because I can't remember quite what Donald Rumsfeld said, but, we try and know all the known problems we have, but there are unknowns which we don't know. And we obviously watch and look for all of these, but we don't actually know what could happen. We've tried to think of every possible scenario and all of the safety issues, but you don't necessarily know. And you can sort of see that, I don't think this is true for these type of cells, but you could see with COVID vaccines, for example, once you start getting up to large numbers of patients, you can start to see some slightly odd side effects which you could not predict. So, in the UK, there was one vaccine in particular. It started causing occasional clotting problems in patients, but you had to be up to thousands and tens of thousands of patients. And to be quite honest, if there's a 1 in 10,000 risk, I think most people would take that. And for most drugs you take, there is a 1 in 10,000 risk for things to go wrong. So it's always the case, you can take any drug and have a bad reaction to it. So as far as we know, we've minimized the risks and we think they're safe. But that is obviously what the regulators are very worried about. And that's why we have very stringent follow-up on all of these patients to make sure nothing happens that we don't know about.
And so what makes someone a good candidate to respond well to cell replacement therapy generally?
So I suspect that the people who are going to do best with cell-based therapies, dopamine cell-based therapies, are the people who have clearly done extremely well with dopamine drugs. So that's another common email I'll get. Can you help me? Can I have one of your dopamine cell therapies? My drugs don't seem to do anything for me. Please, can I have this? Well, if your drugs do nothing for you, a dopamine cell won't do anything for you, because all the dopamine cell is it's a better way of delivering dopamine into the brain at the site where you need it. So if your drugs make no difference, then probably these cells are not going to make any difference to you. There may be reasons why you're not responding to drugs outside of that. But in general, what the patients who are going to do best tend to be younger, I would imagine. They tend to be people who have a very clear response to the dopamine drugs. And then obviously, if they have complications from Parkinson's, which lie outside the dopamine system, so sadly people have developed a dementia or they've got major problems in the so-called autonomic nervous system, the thing that controls your blood pressure, then these cells aren't going to help, because that's not really what the problem is. So the best patients will be people who don't have major non-dopamine complications, respond very well to drugs, and tend to be young and don't have a lot of other pathologies. Those are probably going to be ideal to begin with. Obviously, if the therapy proved to be very effective and very safe, then the envelope, the people you could bring in, starts to expand. But I imagine it's the type of people who are the ones who are up for deep brain stimulation, people who have these infusion therapies. I suspect it will all be that type of patient. And the other thing to say is that people always think of these things in isolation. Well, there's obviously a huge amount of work that's going on, which I know isn't relevant for our podcast chat today, about disease-modifying therapies, things that slow down or stop the disease. There's absolutely no reason to think you couldn't use both of these together. So, in the future, you say, well, I've got a cocktail of drugs that I think could slow it down. Well, if it works, then we don't need cell replacement because you've solved the problem. But assuming it helps but doesn't stop it, you could say, well, I'll repair that bit of the brain, put you on a cocktail of drugs to slow it down, and combination therapies is all you need, and that will sort you out.
Yeah, I was just going to ask him, so what is known about cell replacement therapy used in tandem with other medications or treatments like DBS, focused ultrasound, Duopa, things like that?
It's a very good question, and we don't really know is the honest answer, I would say. So what we can say is that, these cell therapies, there's no adverse effect from the drugs. So it's not as though the dopamine drugs you take have an adverse effect on the transplant. In fact, there's probably a synergism between those. They probably work quite well together. Obviously, if the transplant doesn't work and your Parkinson's continues, this is based on the fetal dopamine cells, you end up having DBS or focused ultrasound or infusion therapies, and that's fine. And currently, for the trials of stem cell-derived dopamine cells, you can't have had previous neurosurgery. So you can't have had deep brain stimulation. You can't have had focused ultrasound. Now, the reason for that is we don't really know what the interaction is between a stimulator and a stem cell transplant. I should think none. But the regulators will want to know that that is the case. It also makes neurosurgery a bit more complicated, even though you're going to different targets in the brain, because someone's already been there and you're going back in with your cells. But there's no reason to think that you couldn't use things like infusion therapies with your cell transplant. So for example, in my clinic, what I'll tell patients, if they're looking at this, I'd say, if you're really interested in cell therapies, I don't quite know whether they'll ever come off. I don't know when they'll come off. But if you have deep brain stimulation at the moment, you'll be outside of that. So if you're really keen on it, let's get you on some apomorphine infusion or duodopa or produodopa, one of these infusion therapies, keep you on that. And then if the cells become available, we can do the transplant, then we can wean you off those other therapies. I mean, people have had cell therapies and then gone on to have deep brain stimulation. That's fine, but you can't at the moment have it the other way around. But there's no reason to think that these drugs would be a problem, or these interactions between these different therapeutic approaches in the future, really.
Might cell replacement therapy be used for something besides dopamine to help people with Parkinson's?
Yeah, so I mean, people often say, well, if this all works, Roger, what do we need to do next? Well, of course, if it works fantastically, there's nothing else. We can all go home. That's the end of it. But let's assume it doesn't. So I think there are two ways in which you could take this forward in terms of therapy. One is you could make your dopamine cells better. So it's fair to say that obviously the pathology which lies at the heart of Parkinson's, which is this alpha-synuclein pathology, curiously does seem to end up in your transplants, at least in fetal dopamine cells. So people are quite interested in engineering stem cells, which is one of their advantages because you can engineer them so they don't express this protein, so they can never develop the pathology of Parkinson's. So you could make a dopamine cell therapy that's resistant to disease. So they could never die from Parkinson's once you put them in, which would obviously be fantastic. The question is that really needed? And I'm less convinced about that. The other way of thinking about it is, are there other chemical systems, are there other neurotransmitters in the brain which are affected in Parkinson's, which could be amenable to cell replacement? And the one which instantly jumps to mind is a system called the cholinergic system. So to explain what this means, so in Parkinson's, you lose the dopamine cells. They sit at the top of the bottom of the stem of the brain, and then they project up. So from a small little site, they send out fibers over a large area. And if you put where those fibers go, or ideally, if you could put them where the cells are lost, then you can rebuild that pathway. And that's how dopamine cell therapy has been used. The cholinergic system, which sits in a different bit of the brain, sits at the bottom of the brain, a bit like the dopamine cells. It projects up into areas, particularly the back of the brain and the outside in the posterior cortex. And we know in Parkinson's that there are a lot of problems in that system. So the loss of that chemical leads to thinking problems. It leads to, in part, to hallucinations. And we know that because when patients develop it in the clinic, we give them drugs which are so-called cholinesterase inhibitors. So they increase the level of that chemical. And certainly for a number of years, in my experience, a lot of patients do quite well. Hallucinations disappear. Some of the other problems they have with thinking disappear. One of the areas which we're currently exploring is, can we make cholinergic nerve cells, like we have dopaminergic nerve cells, transplant them in. They can then innovate that, and then we can treat the dopamine system with our dopamine cells and the cholinergic system. And that would help some of the thinking and some of the psychiatric problems you see in Parkinson's. And so you could think about combining these. So there's a lot of excitement and there's a lot of potential. At the moment, we have to prove that the dopamine cells work. But if that will transform what we can do in Parkinson's.
And so if people are interested in this therapy, how can they access it? Or if they're interested in a clinical trial, is there any advice that you would give to people that are interested in learning more?
Yeah, I think it's terrific that people are interested. So I think there's a great resource, which is the International Society for Stem Cell Research, the ISSCR. So I have got a lot to do with that. They obviously work on stem cells in every area, but they have a lot of very useful information on their website about questions you should ask yourself if you're thinking of having a stem cell therapy. Well, for any condition, but particularly you need to think about it with Parkinson's because people are obviously scanning the internet, finding these places and say, why can't I go there? And I think if you use these guidelines, you can quickly see why you wouldn't go there. So I think it's useful to look at the ISSCR because they also broadcast, if you like, where things are going, as well as new developments. The other places I would suggest is the Michael J. Fox in the States, and in the UK, there's a thing called Cure Parkinson's. These are both charities. They're both very good at advertising what's going on in the cell-based field. There's another place which a lot of people go, which is clinicaltrials.gov, which has all clinical trials are registered there. My only word of caution there is that anyone can register a clinical trial there. It doesn't actually mean it's a good clinical trial. And might not be something you want to have. I think a lot of people, when they see it there, think it must have passed, a whole series of stringent criteria to get there and must be absolutely fine to do. And that's not necessarily the case. So I would say ISSCR, I would say to get yourself into a research clinic or get yourself hooked up to somewhere where they know about it. Always very happy for people to write to me and ask if they want. But these charities are often very good and well informed on this, really. And I'm sure there are other, vehicles by which people can look to it. Final point I want to make about this is stem cell therapy is experimental. They should be subject to a trial. If you are having to pay for it, why? Because you shouldn't be paying for something that's an experiment. So if I've got a bad hip and I want a new hip, I know there's 50, 60 years of research showing that hip replacements make a big difference to a bad hip. If I want to pay for it, I can pay for it, and it makes perfect sense because it's a well-validated, approved therapy. Stem cell therapies are experimental. We do not know if they work. So you should never, ever have to pay for it, or not now anyway. So anything that requires you to cough up some money, don't do it. Go to these other sites and try and get yourself attached to them, and just look out for opportunities to register for things. So things like the Fox, I think you can often register for trials and things like that.
Can you say more about that? Because you do sometimes on TV see advertisements for stem cell therapy with a cost associated, or to travel to get these kinds of therapies. Can you say more about that and how people should have caution?
So, of course, we never get that in the UK, but I know it's very popular in the US. I mean, you just need to be very cautious with things. So most of them are completely innocent. And so at one point I had to review a paper a number of years ago where about 40 patients with Parkinson's had stem cells injected under the skin of their tummy. And what did I think of that in the paper? Well, I mean, there's absolutely no way a stem cell injected under your skin in your tummy is going to have any effect on your brain, as far as I can see. But is it going to be safe? Yeah, apart from a bit of a bruise, you'll be absolutely fine. I mean, the major hit will be your bank account. The point is that people are desperate. So people are exploited. So, and it's worse in some other conditions. So, I used to look after a lot of people with motor neuron disease, Lou Gehrig disease. People would say, what's coming? And you'd say, and they say, yeah, but I've got 2 years to live. Is there any chance this could work? And of course, I couldn't say it couldn't. I said, well, there is a chance. And you say, well, there's some chance versus no chance. So they sort of slightly play on that. So, of course, you're very desperate to have something rather than just listen to Roger say it's 5 years down the line and, another 5 years, it'll be another 5 years and I'll never get it. So people are desperate. They are exploited by agencies and companies trying to sell them what is in essence your sort of modern snake oil. But you just need to interrogate what is the rationale for it? What's the basis for it? What's the evidence that this is work? Has this been published? Don't be taken in by people's testimonies on the website, because obviously they can be quite easily manufactured. And then if I am having to pay for it, why am I paying for it? Because surely, my neurologist or my Medicare company or whatever would have said this is a therapy I should be allowed to have. Anyway, I know it's complicated in the States, but in the UK, if someone came and said, I want a bone marrow transplant, I'd say that's fine. If you've got the right indication, you should have it. Well, the NHS will pay for it. But we wouldn't pay for any stem cell therapy because there's none that's been proven to work. So people need to be cautious. And if it goes wrong, it can go seriously wrong. So there was one of the very early fetal transplant studies. They had it done in China. They injected the cells into the brain. They were not the brain cells. They were other type cells. It grew into a tumor. The patient died. There's a patient in the States who's had a spinal cord injury, had stem cells, now got a spinal cord tumor. Child who went to Russia with a rare neurological condition died of tumors. So these therapies are not innocent. And so you should be very cautious. And, ultimately it's not, you can be very paternalistic and sort of say, well, no, you shouldn't have this. But obviously I don't have the condition and I understand where people are coming from. All I can do is warn them of the dangers of doing it and then get them to think about, is there a rational basis for this? And I always say, if I had, $20,000 to spend on my Parkinson's, would I go and spend it on a stem cell therapy? No, I'd get myself a personal trainer.
If this treatment proves to be beneficial, so what do you think is an optimistic timeline for approval of broad use of this kind of treatment?
So it's a good question. I don't really know the answer to that, but the big companies are basically planning their trials for next year, as far as I know. And what's a bit unclear is exactly what the regulatory authorities will accept as enough evidence to allow it to be used in the clinic. So the standard route is, of course, if I want to do, let's go through drugs for busting clots for heart attack. So, I would do a few dozen patients, check it's safe, show that it works. Then to prove it really works, I need to do a trial of thousands of patients to show that it's safe and it really works. Now, obviously, you can't do that with sticking cells in someone's head. You just simply don't have the capacity and the sort of ethical mandate, if you like, to take hundreds of patients and subject them to this. So the trials are going to be much smaller. And then the question is, if I do a study of 30, 40 patients and I show a very clear benefit, restoration of dopamine in the brain on scanning, they look normal. Is that sufficient for me to go to market with that? And I suspect there's going to be a move towards that in much the same as with a lot of these new experimental therapies. So if that is the case, then within 5 years, the regulatory authorities will have had to make a call on, as will the companies, is this a marketable therapy and should be a standard of care, or is it not working? And the other question which will arise is, what will be the price tag if it works? Will it be competitive in terms of what it's priced at? And that's obviously something of which I have absolutely no control at all. So I think the advantage of having companies involved is they have the capabilities, the capacity and the money to move it quickly. The disadvantage of companies, if there is one, is that they obviously have to wait a while for these cells to work, which is not something they're used to. So, you take a tablet for your blood pressure. You don't want to wait 3 years to see if my blood pressure is lower. You want to know whether it's better in 3 weeks. But these cells you can't get them to work any quicker than that. And also, as with any company, someone can take over as the chief executive and say, don't like cell therapies, don't think there's any money in that, we'll shut that program down, regardless of where that program is. So there is always the issue of, how committed they are. I think they are because they've invested a lot in it. But I do feel this is an area which is likely to deliver one way or another within 5 years.
Is there anything else you'd like to say about what you see as the future of this kind of therapy?
So I think there are two things to say. So often people talk about disease modification. So can we actually slow down Parkinson's and make people live longer with it? Well, that would be a great thing to do, but L-DOPA did that. So, if you had Parkinson's in the 1940s and 50s, you'd probably be dead in 10 years. But L-DOPA actually, in the population level, makes you live a normal life expectancy with Parkinson's, based on, studies we did here, for example. So symptomatic drugs can actually make a huge difference. Most people with asthma are never cured of asthma. But now with inhalers and such like, people live pretty normal lives. So the first thing is, I think, if you've got a better symptomatic therapy, it could have a major benefit to patients way beyond just a better symptomatic treatment. It'll give them a quality of life and a duration of life, which is very different to what you have now. And ultimately, if it works and it's not very expensive, it could be first-line treatment. So you could pitch up at your doctors. You sort of say, I think I've got Parkinson's. You do a scan, you're down in dopamine. We'll bring you in, chuck a bit of dopamine at you. Do you respond? Yep, great response to it. Right, give you a transplant. Don't worry about the tablets. 2 or 3 years, you've only got very mild problems. After that, the transplant kicks in. No tablets. 10, 15, 20 years. Other things might start to catch up with you, but you've had 10 years without anything, without any of the complications which the drugs give you in terms of these involuntary movements, dyskinesia, some of the thinking problems, some of the behavioural problems. So I feel that if this therapy really works, it's not just a better way of giving dopamine. It would transform the landscape for treated Parkinson's.
Is there anything else you'd like to say about this topic? Any other questions you wish I'd asked?
No, I don't think so. I think we covered most things very, very nicely.
Well, thank you so much for joining us today. This has been just a fascinating topic to learn more about.
Thank you.
Thank you for joining us for this episode of the Parkinson's Podcast. For more information about the Davis-Phinney Foundation and to learn about educational offerings and community events for people affected by Parkinson's, please visit davisfinneyfoundation.org or dpf.org. This podcast includes information about Parkinson's and insights from our Parkinson's community. It is not intended as a substitute for treatment advice from your own medical providers.